Welcome to Biotanemy.
Hi, this is a continuation of DNA damage and repair mechanism. If you guys don't see the previous video, the link is given in the icon and also in the description box. Additionally, please consider subscribing to our channel. So, let's move on to the video. Today, in this video, we'll look into mismatch repair, homologous recombination repair, non-homologous recombination repair, and also translesion synthesis. Mismatch repair. Mismatch repair is a post-replicative DNA repair mechanism that corrects base mismatches and insertion, deletion loops that escape the proofreading activity of DNA polymerases during replication. The protein MUD-S homolog 2 or MSH2 forms heterodimers with MSH6 or MSH3 accordingly to form MUD-S alpha or MUD-S beta, which scan the DNA to recognize single base mismatches or insertion deletion loops. After recognition, MUD-L homologs, like MUD-L alpha, are recruited. This complex acts as a molecular matchmaker that coordinates excision and resynthesis. The strand discrimination occurs next by identifying presence of nicks or unligated Okazaki fragments, like replication, associated nicks in eukaryotes, while in prokaryotes it relies on methylation, where the old strand is methylated. The exonuclease, EXO1, will remove a segment of the new strand containing the mismatch, which spans hundreds of nucleotides. The DNA polymerase delta or epsilon will fill in the gap using the correct template. DNA ligase 1 will finally seal the nick to restore the DNA backbone. Homologous recombination repair, predominantly active during the S and G2 phases of the cell cycle, when a sister chromatid is available as a template. It repairs the double-stranded break that occurs due to various factors. The broken DNA ends are first recognized by damage-sensing proteins, or the mRNA complex, which binds to the DSB and initiates end resection, creating 3' single-stranded DNA overhangs. The single-strand DNA overhangs are coated by RPA in order to prevent secondary structures and degradation. The RPA is then replaced by R8051 with the help of BRCA1 and BRCA2 proteins. R8051, a recombinase, forms a nucleoprotein filament on the ssDNA. This filament invades a homologous region on the sister chromatid, forming a D-loop. The undamaged sister chromatid serves as a template to copy accurate genetic information. DNA polymerase extends a 3' end of the invading strand using the template. Finally, the complex structures like double holiday junction or displacement loops are cleaved by specific resolvase enzymes and helicases. Homologous repair can also occur through the subpathway synthesis-dependent strand annealing without forming a double holiday junction. The structure is resolved, restoring an intact and accurate DNA sequence. Non-homologous end repair. Non-homologous end joining will repair the double-strand breaks that occur due to various factors. The double-strand break, recognized by the Ku80, Ku80 heterodimer, binds to the DNA ends, which protects the ends from degradation and recruits other NHEJ factors. Next, the DNA-dependent protein kinase catalytic subunit binds to Ku-bound DNA to form the DNA-PK homoenzyme, which brings the ends of DNA closer, to form a synapse. Now, these DNA ends are often incompatible, like they are blunt, have overhangs, or are damaged. So, several enzymes will process the ends, such as enzyme Artemis is an endonuclease that will trim overhangs. Polynucleotide kinase phosphatase will modify the DNA ends to make them ligatable. Then, the enzyme TDP1 APTX will also act depending on the damage. Nucleases or ligases will perform the final phosphodiester bond formation between processed DNA ends. Now, let's also talk about translesion synthesis. Translesion synthesis is a DNA damage tolerance process that allows DNA replication to continue past lesions that would otherwise stall the replication fork. It is not a repair process per se, but rather a damage bypass mechanism. It fixes bulky or helix-distorting DNA lesions that block high-fidelity DNA polymerases. Translesion synthesis happens during S phase, when the replication fork encounters DNA damage, and is triggered when replicative DNA polymerases stall at the lesion. Specialized TLS polymerases can accommodate distorted templates and insert nucleotides opposite damage bases are 1. Ku80 bypasses UV-induced thymine dimers efficiently. Defects cause xeroderma pigmentosum variant. 2. Polyota bypasses minor groove lesions, though error-prone. 3. Pol-kappa bypasses bulky adducts. 4. Rev1 insert cytosine opposite of basic sites, or G, acts as a scaffold. 5. Pol-zeta extends synthesis beyond the lesion after insertion by other TLS polymerases. High-fidelity DNA polymerases cannot synthesize across the lesion, so when they encounter the lesion, they will stall. At that time, the Rad6, Rad18 complex will monobiquitinate the pCNA protein at lysine 164. The stalled replicative polymerase dissociates. A TLS polymerase is recruited to insert a nucleotide opposite the lesion. Another TLS polymerase may extend the chain.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.